Okay, for our experiment, we did the nutrient effects on overall water clarity, and that was with Jaden and me. We did that on bodies of water in Phoenix. Our overall objective and goal for this research was to just test the different types of nutrients in bodies of water and to see what kind of effects they were having on the clarity and how those all correlate together. Our research question was to ask what is the effect of the nutrients such as pH, phosphorus, and nitrogen, and also KH on water clarity. And some things about why water clarity is actually just important in general is that nutrients are really crucial for plant growth and algae, and that's really important because they are at the beginning of the food chain. Similarly, phytoplankton is also there, so that's why clear water is really important. That way, the sunlight can actually reach those phytoplankton and help feed them, and then they can go on to feed other animals in the food chain. And then if we have too many nutrients in these bodies of waters, it can actually cloud the water and make it harder for sunlight to reach in, and those photosynthesis processes aren't going to occur as well, and they're going to be harder to complete. Our hypothesis was that as the nutrients of the water decrease, the overall clarity will increase. Our reasoning behind this was that just as higher nutrient levels often encourage the growth of algae, it reduces clarity. That's because of the eutrophication. So again, that sunlight can't reach those organisms and they can't clear up the water and there's not oxygen and it just kind of becomes a really toxic environment for a lot of animals and organisms in those bodies of water. Here is just a little bit of our experimental design. So our variables, our control was our tap water. We just got that from inside of the lab. Our independents were our pH, nitrogen, phosphorus/phosphate. We did about 5 samples and then one of them outside of that, so 6. One of those was the control, which is the tap water. So we had 5 different samples from around Phoenix. And then our experimental design, we just had our water clarity measured using the spectrophotometer. And then our experiment, we tried to keep as controlled as possible. The timeline was a little bit unfavorable, but we really just tried to use gloves and do everything at the same time that we could. And then these are just a couple photos of us sampling. So the first one is Jaden sampling at Papago Park. The second one is me sampling at Granada Park. And then the last photo is where we exactly sampled on the IMAPs of Encanto Park.
For the treatments of the samples, gloves were used for treating the during the treatment of the water samples, just to make sure that any contamination didn't occur in the water samples, to try to limit the errors. And then the methods that we used absorbents to better record the turbidity in each of the waters, and that was measured in nanometers. Absorbance is a measurement of how much light is absorbed by a substance at a specific wavelength. So the duration of the experiment was over about 3 weeks. We ended up collecting water samples on November 2nd at each of the 5 spots. And then spectrophotometry was used on November 13th to measure absorbance of the samples. And then we ended up getting the test kits later on. And so November 20th was when we tested nitrate, pH, the hardness, and phosphate. So for data collection, we collected the pH level, the hardness, nitrate, phosphate, and absorbance levels. We went to each of the test sites and collected data samples in clean plastic water bottles, which were labeled with duct tape and placed in a fridge after each of the sites were tested. And then data was collected on a spectrophotometer a week after that, and then a week after that, test strips were used to take samples for the pH, hardness, nitrate, and phosphate. There were no really abnormal notes, just some of the water samples were more clear than others. And then here are our results from our raw data, and so Nothing really stands out besides the hardness in Encanto Park. Um, and the pH levels all seem to be very similar except for Granada and relatively close Tempe Lake, but mostly Granada. And then absorbances had some variance. So tap water had the least absorbance, and then Steel Indian had the highest absorbance. All right, and here is a data graph, or a bar graph with all of our data. There was no direct correlation between any of the points, which was really disappointing to see, but there could be a correlation between the hardness of the water and the absorbance levels and turbidity, but there's just the higher outlier with Encanto, and then the lower point in Steel that seems to be across the board. And so a conclusion cannot be reached, and no other assumptions can really be made except that because, yeah, the pH level stayed relatively constant. Other data is generally uniform with little variation. So no true correlation there from what can be seen from the data. Okay, so we took t-tests for all of the separate data points, and so for phosphate, phosphate shows it's a nutrient that, if in excess, can cause a lot of bacterial growth and cyanobacterial growth, which is like algae and stuff. The TNP scores were very similar, which means that there's no direct like correlation. We used 95% confidence interval for each of the tests. And then going to nitrate, it just shows the amount of nitrogen materials in the water, and that also has to do with like excess growth of different bacteria and stuff. If there's too much, it also produces a lot of algae. And then T-score and P-score values were very similar. So no direct correlation there either. And then same as KH, which shows the general hardness of the water. And Arizona has a really high hardness level across the board. So it was expected to have not much of a difference between water tests. KH is potassium hydride, which isn't super relevant to this specifically, but it does help to show variances in water quality. And then pH did seem to have a surprising, statistical relevance. The pH of a liquid shows whether it is more alkaline or acidic, which can have an effect on marine life. And the T-score value was 29.1, which is actually very relevant because the p-value was also 1, which if the t-score is higher than the p-value, there's an assumption that the test is very compatible with the data. So there is something significant with that, which is really promising, but because our data was collected at such a late time, I'm not sure if we can actually determine that this is statistically relevant. So there's no significant correlation except possible significance in pH. We wish we could have collected the samples and performed the tests sooner, but we could not because we did not have the testing materials just at the proper time. We also didn't have proper vials to collect the samples. I was not able to get them, and so we had to use empty water bottles. They were clean, they were not used. The water was just poured out of them, so we tried to keep it mostly controlled, but it's very unclear. So the results were kind of both expected and unexpected, specifically with pH. So the null hypothesis in this instance is rejected because the results did not align with what was expected. And I would definitely do the data collection differently if we performed this experiment again.
Some mechanisms and ecological implications. A lot of the mechanisms are just really similar. So the higher and more increased that they get, they're going to increase and support algae growth. So more algae growth means more algal blooms. And again, as this whole experiment has been talking about, just reducing the ability of sunlight to penetrate that water because of the algae suspended particles on the top that float around, it's really hard for that sunlight to reach those aquatic plants and organisms that need that to photosynthesize or make food. And so they are really at the beginning of the food chain, the plants and the phytoplankton, for example. And so that can kind of just throw off the whole ecosystem in itself. And so pH, nitrate, phosphate, and KH, which is going to be our carbonate hardness, those all really increase and enhance algae growth. A couple of implications are going to be the algal blooms. Again, these just really block the sunlight and decrease the water clarity. They also give a little bit of a green look to the water, and that's why some places may look greener than others, just depending on the amount of algae. And algae really just leads to eutrophication. So oxygen will deplete, and this affects all aquatic life because plants need that oxygen and other organisms like fish. They really need that to survive. And if the bacteria that is helping decompose this algae that keeps dying and going to the bottom, it uses all this oxygen up and there becomes none left for the rest. And that becomes a problem because it disrupts the food web because phytoplankton and aquatic plants are at the beginning. It has a large effect on the entirety of the rest of the food chain because they are at the start of it. So it's almost just like a domino effect. So now on to our conclusion. Our hypothesis was not supported a little bit, maybe because of the errors in our timeline. It was not favorable again. But that's okay. So it's not supported because as the nutrients of the water decreased, the overall clarity did not increase. And the effects of the treatment on the conclusion, they're all a little bit similar, just back in the mechanisms. They have the same effect on algae. They're going to promote algae growth and are going to end up decreasing the overall water clarity. So a little bit of the broader impacts with these is again, they promote the algal blooms. So these blooms are going to give the water a really big greenish color and are going to stop and hinder that light from penetrating through there. Again, back to the eutrophication with the hypoxia and toxins. The bacteria really use up that oxygen that a lot of aquatic organisms need to live, and this creates hypoxic environments for these organisms, and it really stresses them and can make the water toxic for them to live in. And populations may start to die off. With this, algae clarity decreases and water will not be as see-through. And a little bit to manage the pollution, we could create some buffer zones. So, kind of like the wetlands around these bodies of water to really have areas to absorb those nutrients. That way, they're not all going into these bodies of water where these organisms are living. These nutrients are really good for places on land and again, these wetlands. So, restoring these wetlands are going to be crucial to helping stop the algal blooms from completely destroying ecosystems. And then also implementing a little bit more aquatic plants. These aquatic plants can really help provide more oxygen back into that water. And as long as they're getting that sunlight and having those photosynthesis process that they need to have done, they're going to be a big help to these ecosystems. Okay, and then this is just our resources slide.